Joining today's webinar. My name is Katie Montgomery, and I will be your host. Today is the second webinar in our conversation series, Drug Hunting in a Digital Age. This webinar is titled Design of a Highly Selective Allosteric Picomolar TYK2 Inhibitor in Clinical Development, a Tale of Two Sites and Innovation in Modeling. Our speakers today are Craig Massey and Cyan Mondal. Craig was formerly the head of medicinal chemistry at Nimbus Therapeutics and is now the SVP of discovery at Ajax Therapeutics. And Cyan was a senior director in Shcrodinger Therapeutics Group for many years and is now executive director of computational chemistry at Entos. And I will pass it on to our speakers for the talk.
Thank you, Katie. So the breakthrough by design approach at Nimbus really required a greater commitment to computational methods and for ligand design and optimization than was common in the industry. This approach was really enabled by our integrated partnership with Shcrodinger. So our project teams consisted of Shcrodinger scientists working alongside Nimbus chemists and biologists to produce bespoke code and develop novel in silico algorithms to overcome program-specific challenges. The teams then designed, iterated, and optimized these leads using next-generation physics-based technologies, which allowed us to rapidly refine our discovery methodologies. This partnership provided Nimbus with unparalleled computational horsepower, including some unprecedented, at least at the time, FEP modeling efforts, which Cyan will discuss as part of our presentation today.
To add a personal touch to it, my memories of Jeremy drawing prolific amounts of compounds, putting in a lot of ideas with both enumeration and hand-drawn designs that we would model and sit around live design and discuss the data with. And then Craig would go after the most promising compounds, no matter how challenging the chemistry. And so we worked very close together day in, day out. And also on the Nimbus side, there was Wenyan on the biology side, who always asked for more potency, and Heather on the ADME DMPK, who asked for everything else. So with those goals in mind, the team worked with modeling across many different parameters and at scale to achieve the final compound.
So TYK2 is a member of the JAK family of receptor-associated kinases, which has emerged as an important immunokinase target. So IL-12 signaling, which is shown here on the left-hand side of the slide, requires TYK2 kinase activity for effector function, which drives differentiation of Th1 cells that are associated with the TNF-alpha and interferon gamma pathogenesis. Likewise, IL-23 signaling also requires TYK2 kinase activity, which drives differentiation of Th17 cells that are associated with the IL-17 and IL-22 pathogenesis. Both of these pathways are strongly associated with the psoriasis, Crohn's disease, and ankylosing spondylitis pathologies. TYK2 is also involved in type 1 interferon signaling, shown here on the right-hand side of the slide, where it activates multiple inflammatory cell types and is associated with the lupus pathology. Additionally, there was also highly compelling genetic evidence in the form of GWA and PheWA studies for a role of TYK2 in a number of autoimmune diseases. But selectively drugging TYK2 is challenging due to its high homology with the other JAK family members, namely JAK1, 2, and 3. This figure illustrates the various domains of the TYK2 protein. The team's early efforts actually focused on the development of catalytic domain or JH1 inhibitors, using FEP modeling to design in selectivity versus the other JAK family JH1 domains. This allowed the team to identify a candidate that was sparing for JAK2 and JAK3, but still displayed appreciable activity on JAK1. To overcome the selectivity issue, the team initially investigated the development of dual domain JH1, JH2 inhibitors by modification of our catalytic domain inhibitors. However, this approach was not well structurally enabled. There was only one dual domain crystal structure known, and it lacked suitable resolution for FEP modeling. Fortunately, around this time frame, the team came across a number of references from BMS, which we'll discuss a bit later in the presentation, which clearly demonstrated that TYK2 pseudokinase or JH2 binders could functionally inhibit TYK2 signaling pathways. Given the deep structural enablement that was available for TYK2 JH2 domain, we believe we could leverage the large-scale FEP modeling that we developed for the catalytic inhibitor program to rapidly identify highly selective TYK2 JH2 candidates.
So like Craig mentioned, as a prelude to going after the allosteric binding site, we actually developed a lot of the methodologies and approaches while working on the JH1 domain. And here you can see the binding site of TYK2 superposed with JAK1, JAK2, JAK3. And frankly, no one can tell them apart. So this provided the background and context to go beyond the typical medchem strategies of docking the compounds and looking at the dock poses of many ideas and selecting compounds for potency and other properties, and instead use physics on a very large scale in order to differentiate the binding between TYK2 and the JAKs, JAK1, JAK2, JAK3. So for that, we used FEP to predict the potency on target in TYK2, as well as to predict the potency off target on JAK2 and JAK3, and then pursue the chemical targets that were predicted to give a good balance of potency and selectivity. So in FEP, the fundamental point is that we can take into account with rigorous physics both the ligand in solvent as well as the ligand bound to the complex, taking into account the flexibility and dynamics of the protein around it. So we can taking into account both these lines of contribution for both the enthalpy and entropic contribution, we can then figure out what is the binding potency for the ligand to the protein of interest. Alongside potency prediction, we also wanted to have modeling to keep the properties in line and solve challenges on the ADME side. So one of the another innovation in this project was the introduction of computing solubility with FEP. So in this figure, you can see a thermodynamic cycle of a bunch of ligands forming an amorphous aggregate, which approximately corresponds to what the state might be, solid state might be in kinetic solubility measurement. We take one of the ligands from the interface area into vacuum via FEP using total annihilation of the ligand with FEP, and then go from vacuum into the water. In these two steps, we are able to compute what is the free energy of taking a ligand from this aggregate into water. And we can do, because it's a disordered system, we can, for at least amorphous state, we do this for a bunch of different ligands and then take the median of those. The main point of this approach is that not only can we move solubility by increasing polarity, but also by breaking also via the lattice energy by breaking and destabilizing the aggregate form. And you will see an example shortly. So for the JH1 project, we had several promising compounds before deploying FEP. However, although we had several potent compounds, they were always missing a parameter in either JAK2 selectivity or JAK3 selectivity, and in some cases solubility as well. And the major goal here was to find compounds that are very potent in the picomolar regime, as well as have close to 2 log order selectivity against JAK2 and JAK3, as well as being soluble. So this is, faced with this challenge, we debuted what was then something that has never been done or even envisioned before, which was to use FEP on the scale of thousands of ideas and to inform synthesis on a daily basis with the help of modeling with rigorous physics, potency, selectivity, solubility. So we used a funnel like this where we enumerated a few thousand high quality molecules. Now these are all have reasonable poses, so they've all passed MMGBSA filter as well as reasonable phys-chem properties. Then we calculated the potency on target, and then compounds that were predicted to be potent were passed on to JAK2, JAK3 modeling, and finally solubility modeling, and then 46 compounds out of this 4,000 compounds were targeted for synthesis, of which 9 of them met every single criteria. So we went from struggling to meet criteria for across the selectivity, potency, solubility spectrum to having multiple breakthrough compounds with this. In the course of this, there were several small pieces that we figured out in order to make large-scale FEP feasible. That includes how to sample R groups around torsional barriers. When we annihilate asymmetric R groups, we want them to flip around and sample both sides of it, for example. We debuted a strategy that combines single-edge quick scan of potency, followed by a more rigorous cycle closure scan with error bars and so on. We deployed FEP in the Amazon cloud for the first time at scale. And of course, we just talked about the solubility FEP as well. So the entire computational effort was completely unprecedented in drug discovery at that point, and consisted of 250K GPU hours for this exercise. And out of this, we came up with a bunch of compounds that where each can be considered to be a breakthrough in terms of balancing potency, selectivity, solubility. To give an example of getting solubility without taking recourse to polarity, which can break other things like permeability, this is an example where this R group, when we change it to these two in the solvent exposed area of the pocket, we are not changing in this example log P or PSA that much. It's basically in the same regime. If anything, the log P is becoming worse, slightly more hydrophobic in this case. Nonetheless, FEP solubility correctly predicts that we are able to move the bar from single-digit micromolar solubility to reasonable solubility to progress. And this is borne out in both kinetic solubility as well as thermodynamic solubility, where we see improvements for these two compounds, even though the PSA and log P aren't really changing that much to give us solubility. And we can do all this maintaining the picomolar potency as is shown here. With these methods, we were able to quickly achieve breakthrough after breakthrough and make excellent progress on the JH1 domain to development candidate compound. However, then we had to make a pivot. Greg?
Thanks, Sayan. So this slide highlights the two BMS publications that I mentioned a bit earlier in the presentation. The original Journal of Biological Chemistry paper shown here in the top involved a chemogenomic screen of a BMS kinase inhibitor library using an IL-23 TYK2 cellular assay. This resulted in the identification of the imidazole thiazolopyridines, shown here by structure 3, as a domain-selective TYK2 JH2 binder that displayed promising activity in the TYK2 cellular assay. Now, importantly, this was the first demonstration that TYK2 signal transduction could be inhibited by binding to the pseudokinase domain. So it was really effectively true allosteric inhibition of TYK2 kinase activity. Now, optimization of scaffold 3 proved to be difficult, owing to some off-target selectivity issues with IKK beta. But a subsequent MedChem communications paper, shown here at the bottom of the slide, also from BMS, revealed that their phenotypic screen also identified this imidazole pyridazine core, shown here as structure 7, which displayed improved kinome selectivity. So the combination of the biological validation for allosteric inhibition provided by the BMS publication and the deep structural enablement available for TYK2 and the other JAK family member pseudokinase domains provided the team really with a perfect opportunity to engage in a multi-pronged structure-based drug design approach using the full complement of Shcrodinger tools. So this slide captures the diversity of chemical matter from each of these structure-based design strategies. On the left-hand side, you'll see an FEP core hopping effort from some of the patent chemotypes. This is from the BMS patents. This led to the identification of the pyrazolonopyridine and pyridinoimidazole cores 1 and 2. Now, while these cores displayed very promising potency and domain selectivity, several ADME liabilities prevented their rapid advancement. On the right-hand side of the slide, it represents a virtual screen workflow in which a large library of lead-like and drug-like compounds, in this case from an E-molecules library, were screened against an ensemble of receptor models based on several known JH2 co-crystal structures from the PDB. So this effort resulted in the identification of 3 structurally distinct hit series, exemplified by 3, 4, and 5 here. Now, while all of these hit series displayed some promising levels of JH2 affinity, we were especially intrigued by the steric and electronic similarity of the pyrazolopyrimidine core to the imidazopyridazine core that was just published by BMS in their MedChem communication paper. And overall, the team was quite pleased to find that these structure-based design efforts allowed us to identify multiple picomolar JH2 scaffolds from these hits within a period of about 3 months.
So in order to progress these hits, we employed the multi-parameter optimization paradigm with large-scale modeling that we had debuted for the JH1, and we did it in 2 stages, basically. The first stage was employing quick modeling of Phys-Chem properties as well as permeability, passive permeability, in order to make sure the compounds that would go into the resource-intensive FEP models as well as synthesis would stay in a reasonable chemical space. And this was particularly important, for example, for the pyrazolopyrimidine series, because most of the compounds there were in the PSA greater than 120 regime, so around here, and therefore it was important to keep their permeability in a good space. So for this, we used this quick MPO function where each of these property was put together in a geometric mean. To do that, each of the property is projected onto a 0 to 1 scale and with soft cutoffs that define the boundary regions. The really important piece here is, number one, that we use soft cutoffs here. There is an error bar associated with both the choice of cutoffs as well as the models, as well as the assays and so on. So the soft cutoffs make sure that we can take into account multiple different parameters together without losing compounds that will just miss a cutoff by just a little bit. And the second piece is that the final decision is sensitive mostly to where it ultimately matters for the final end result. So for example, if the permeability is predicted to be super good here along this versus simply good, it does not really affect the decision that much, and you take into account other aspects of modeling by then. Similarly at the BAT regime. So with this sort of framework for decision making, we were able to, for example, find a lot of permeable compounds in the pyrazolopyrimidine series, even though we were in the PSA 120 regime. Here, for example, in the pie chart, it shows that compounds that were made with RRCK in the yellow zone were mostly permeable at about 80% and higher rate. Maintaining the compounds in the good property space, we aggressively tried to improve the potency or keep it in the picomolar regime. So in this plot, you can see that most of the compounds, at least for the iotomers that we made, were predicted and measured to be picomolar, super potent. And about 80% of the compounds were measured to be within 10-fold of the potency that was predicted with FEP. So after passing compounds through the quick MPO filter, we could put them through this rigorous FEP potency predictions and put together a full modeling MPO. So we were targeting picomolar potency, So either double-digit picomolar potency, which was the target, or at least triple-digit picomolar regime. We put together all this, all the models and MPOs in live design in different live reports for each design objective, and the team would together go over this. So this is an illustrative live report where you can see that the total live report contains about 1,000 ideas that were scored across the board, but the top 3 ideas out of that are looking promising here, with promising potency, solubility, RRCK, PSA, and log-P, and were selected for route scouting by Craig and the MedChem team.
So the team adopted this MPO strategy that Sayan just walked you through, and we carried out parallel ligand FEP modeling against TYK2-JH2, as well as the other JAK family kinase domains, in order to design analogs with high JH2 affinity and JAK family domain selectivity. So we conducted a scaffold hopping exercise from compound 6 in this table that involved adding another hinge binding element at the C5 and C7 positions of this scaffold, and this was based on similarities to some of the BMS chemotypes. We were pleased to see that compound 8 displayed double-digit picomolar JH2 affinity and good TYK2 cellular activity. However, compound 8 still displayed potent activity on PDE40, which is something we expected based on the MedChem report from BMS. Now, FEP-driven optimization of compound 8 led to the identification of compound 10, containing a cyclopropyloxy pyridine replacement for the dimethylphenyl group in compound 8. The choice of the larger substituents on the pyridine was based on a co-crystal structure of compound 9 with TYK2-JH2 that suggested that larger groups would be tolerated within TYK2, but not for PDE4D due to a clash with the zinc binding region in our PDE4D models. Compound 10 displayed JH2 affinity and domain selectivity that was comparable to compound 8, but with much improved selectivity versus PDE4D and some improved metabolic stability. I should also note that all these scaffolds displayed high functional selectivity versus JAK1 through 3 signaling pathways in our human PBMC assays. Compound 10 also displayed a promising rodent PK profile and served as a useful starting point for further optimization of this scaffold. The team next carried out right-hand side chain explorations using a large-scale FEP screening campaign from compound 10 to enhance the potency and domain selectivity of the scaffold. We also conducted more qualitative modeling on PDE40 to further improve the selectivity versus this off-target. Now, a small set of the right-hand side modification is shown here, along with the histograms of the FEP predictions across each of the subseries, to illustrate how we focused our synthetic effort on ideas predicted to deliver picomolar JH2 affinity. Now, while each of these subseries displayed high levels of JH2 affinity and domain selectivity, as was predicted by us on the FEP modeling, we ultimately focused down to the N-arropyridones, exemplified here by compound 15, based on improved cell potency, PDE4D selectivity, and metabolic stability of the subseries. Another important design consideration was that the N-arrow group on the pyridone provided us with an alternate vector from which to enhance selectivity versus PDE4D. This was due to a steric clash with the magnesium binding region in that protein. After conducting several rounds of in silico ADME modeling on the N-arropyridone subseries, the team identified compound 16 as an early lead candidate. And while compound 16 displayed a good potency, selectivity, and ADMET profile, it was determined that it did not possess an adequate human predicted PK profile to support additional progression, and that was based on some allometric scaling work as well as IVIV extrapolation calculations. As such, the team's effort shifted to further optimize the potency, in particular the human whole blood potency, and the human hepatocyte stability of the series. Based on a JH2 co-crystal structure obtained with compound 16, the team believed there was an opportunity to better fill the space in that region of the protein near the C3 amide. So the team then modeled and prepared a variety of amides, a few of which are shown here in this table, and our FEP data suggested that a methoxycyclobutylamide would best fill the space and was predicted to provide an additional protein interaction between the methoxy group on the cyclobutyl ring and the pseudocatalytic lysine in TYK2-JH2. Gratifyingly, compound 21 displayed single-digit picomolar JH2 affinity and improved human whole blood potency, but it did not improve the human hepatocyte stability relative to compound 16. Fortunately, a small modification of compound 21, which involved incorporation of a desfluorpyridine intended to increase aqueous solubility, led to compound 22. This compound was initially known as NDI-034858 and later as TYK279. This compound displayed comparable human whole blood potency to compound 21, with much improved human hepatocyte stability and some increased solubility as well.
So this is a histogram where during the AMIT survey, we were able to figure out a piece that improved the potency by quite a bit. You can see that most of the compounds, most of the ideas scored around 1 nanomolar to triple digit picomolar, and some of these compounds were flagged in FEP to score improved potency by a log order to double digit picomolar. And one of these led, after optimization for the properties as Craig outlined, to the final DC compound. As Craig mentioned, the cyclobutoxy piece interacts with the pseudocatalytic lysine here, and for the TYK2 JH2 domain, but it actually clashes with an isoleucine in JAK1 as well as JAK2, and that is in part responsible for the big selectivity that we get from this piece, as well as being exquisitely potent on target. Together with the exquisite potency on target and the selectivity against the JAK family, we also get exquisite selectivity against the kinome at large. And you can see that there's barely any off-target kinase that the compound hits. To reach this DC overall, we modeled more than 13,000 ideas through full cycle FEP, which is an unheard of number at that time, but it's quite normal these days, across multiple chemical series. And one of the aspects of this program was that we were always generating new chemical series as we went along with the help of our modeling, and trying to quickly reach the end of those new chemical series. Overall, we reached the DC compound in about 2 years from starting the Allosteric program.
So in February this year, Takeda acquired Nimbus Lakshmi, which was the subsidiary of Nimbus containing the TYK2 inhibitor program, including NDI-034858, now known as TAK-279, for $4 billion. The team was obviously incredibly excited to reach this milestone for what we believe is a potentially best-in-class molecule to treat a broad range of autoimmune diseases. About a month later, Takeda reported the phase 2b clinical data of TAK-279 in patients with moderate to severe plaque psoriasis. This is at the AAD meeting. The team was thrilled to see that the highest dose of TAK-279 tested, about half of the patients achieved PASI 90 scores, and roughly a third of patients achieved PASI 100s, which means complete resolution of their skin lesions. Takeda is planning to initiate phase 3 studies in psoriasis this year. Is also evaluating TAK-279 in several other immune-mediated disorders, such as psoriatic arthritis, lupus, and IBD. Of course, the discovery and development of this compound was an incredible team effort involving a large number of dedicated and highly integrated scientists from Shcrodinger and Nimbus. I'd just like to call out a few folks from Nimbus and Shcrodinger on this slide. So on the Nimbus side, I'd like to extend a special thanks to Wenyan Miao, who is the biology lead for the program, as well as Heather Blanchett, who is the DMPK lead for the program. On the Shcrodinger side, I'd like to add a special acknowledgement to Jeremy Greenwood, who, along with Sayan, were the computational chemistry leads for the program. I often referred to Jeremy as the chief ideation officer for the team due to his amazing ability to hand draw designs, thousands of them, to overcome multiple challenges on the program, as Sayan mentioned. The team mourned the loss of Jeremy about a year ago, but we also celebrated his incredible achievements on this program and many others. And then finally, I'd just like to thank all the webinar participants today for your attention.
All right. Thanks, Craig and Cyan, for a really great presentation. We have plenty of time here for Q&A, so I encourage everyone to submit questions, and we'll dig into it. So questions come in. How many ideas did you have to evaluate in FEP to come up with new cores?
Yeah, so answering this more generally than TYK2, usually we evaluate something like a few hundred to 1,000 ideas to come up with new cores. And it's particularly effective if it is done in an iterative process between the modeling and the ideation process. So typically we would draw 50, 100 ideas, and then things look promising, and then we can iterate around that using that as the hook.
Great. Okay. Another question that came in around, did your potency, for example, criteria change over time? Did it get more stringent for different steps in the process? Is a sort of general question.
Yeah, so also answering that more generally, yeah, so the TPP can evolve in response to data, as well as the TPP, we go after that in stages as well. So across projects, again, in general, when we do the initial hit finding, we might just be happy with, in a classification sense, to have a potent code to start with. And then we define MPO based on the TPP, and that MPO can and does evolve with project stages. I don't know if Craig, if you have anything to add.
Yeah, I just say, you know, for the TYK2 allosteric program, I think we knew that we had to be in the low nanomolar or picomolar regime for TYK2 JH2 affinity. We knew that because of the work that is done at BMS, right? That you really needed those high affinity JH2 binders in order to see appreciable cell activity. So I think we had pretty high standards right from the beginning of this program to be in the picomolar regime for our binding assays.
Great. Lots of good questions coming in. Did the FEP protocol for the R group optimization utilize common defaults or custom parameters? Was it customized? And what do we use that? This is, but Cyan is going to really stretch your memory here. But
no, it's as if it happened yesterday. So this is actually a really good question, and we actually came up with many of the technical details to make this all work when we were doing the TYK2. So one thing we wanted to make sure was that the R group can sample both different orientations of the ring. So we annihilated the whole piece and grew it and so on to increase the sampling. Another piece was that we would obviously, before we did anything, we looked at the convergence over time. And based on that, we decided whether to run for 10, 15 nanosecond and so on. So most of the calculations were run much longer than the 5 nanosecond or whatever is the default there. So yeah, so again, going more generally, because everyone will have their own protein target to work with further, I would always begin with a validation exercise that look at how long it takes for the different R group perturbations to converge at different position and be guided by that. But one should expect in general to change from the default parameter for the particular protein.
Great. Is how and is if FEP able to handle cases of scaffold hopping?
Yeah, so this was actually the first program ever where FEP was used to do scaffold hopping, and it actually provided the first proof of concept that this can be done really well. And so we came up with we went after 3 cores, if my memory serves right, out of the gate for the allosteric program using FEP scaffold hopping, and every single one of them was as potent as predicted. And this process has again been replicated across multiple projects since then, and it has always really worked very well.
Great. What stage did you incorporate IP evaluations into the compounds?
Yeah, so we would typically do a lot of, maybe some prototype analogs just to validate the FEP modeling for a scaffold without doing IP. But certainly before we did any large scale and before we went any further with a large scale FEP screening campaign that Sayan spoke about today, we would have an FTO analysis done on those scaffolds to make sure, right, we're going to invest our chemistry efforts in that. We want to make sure we have a path forward in terms of freedom to operate.
Okay. Did the use of predictive modeling impact the amount of synthetic effort going into some of the chemistry and the harder chemistry?
Yeah, certainly. I think that our two main goals with integrating the FEP modeling with the MedChem ideation really was to focus the synthetic effort, right? That was the goal here, is kind of reduce the iterations around the typical design, make, test, analyze cycle, right? So we focus the synthetic efforts on the high value, the high probability of success target molecules, even in the face of challenging chemistry. If it's a challenging scaffold, if it was a high value, high probability of success for picomolar inhibition, we would go after those scaffolds. So it really does allow you to focus the synthetic effort. And equally importantly, if not more importantly, in my book, it allowed for the early elimination of ideas that were unlikely to meet our desired potency or our Jack family selectivity thresholds.
Great. How many compounds did you put into rat PK? What was the cadence of getting in vivo data? Speak to that.
Well, now you're stretching my memory a bit here. Mine's not as good as Sean's. I do not recall how many we put into rat PK, but certainly as we advance compounds down a typical medchem screening cascade, right, and we had things that looked promising and had the ADME profiles that were suitable, we would get them into rodent, either mouse or rat PK studies quickly so that we could evaluate them in our PKPD and then subsequently efficacy models.
Okay. Can you describe what software was used to implement some of the MPO modeling, and how did you decide on the cutoffs?
So we use Live Design, and Katie can tell you a whole lot about Live Design, I'm sure. So Live Design has inbuilt MPO, where it basically you can decide these are the variables of interest, and it computes a geometric mean of the different curves. And in terms of deciding on the criteria, that's guided by the TPP jointly as a team with the med chemists, biologists, modelers, everyone together. And like Craig said, for example, we knew out of the gate we wanted picomolar compounds on this target, so that guided the potency criteria, for example.
Great. Can you describe a bit more about your ADME property modeling and how you did it and what the approaches were?
Yeah, so for the ADME, first of all, we looked at PSA log P, typical things that everyone does, and try to be within a reasonable regime. But then the passive permeability is the first, I would say, interesting modeling that we used. So the passive, there are a couple of references out there, but the way it works is it's a very quick physics-based model that takes from based on dielectric, takes a ligand from water into a hydrophobic media, which is actually represented by chloroform based on the dielectric state. And what the developers have done ahead of time is regress it against all the public data that is there for the RCK model. So there's only a couple of parameters, so we know what those parameters are. Then we use it directly into our chemical series of interest in the program. At the very beginning, it's important to actually obviously look at how it's tracking with the experimental data coming in and figure out what kind of cutoff, if there is any series specific adjustment that needs to be made in the cutoff and so on. So that's for the passive permeability, but a really important piece of it is that it allows for some amount of ligand conformational sampling in. So essentially, if there is any, let's say, internal hydrogen bond that shields a polar moiety, those are taken into account, and those are really important design elements, as we understand. So the passive permeability modeling can be run very fast, and so we put it together with the log P and PSA, into this quick MPO, and that triages compounds that go into much heavier modeling, which are based on FEP. And for FEP, we used FEP potency regularly on target, for example, and you saw example, we talked about that, and that was used both for R groups as well as for scaffold changes. And we also used as needed FEP solubility, and that was a new technique that was developed, and it's based on absolute FEP. So FEP potency in this program is based on relative FEP. So you have reference compounds based on which you compute what the potency of new compounds that make by making changes to that. Solubility FEP is standalone. Each compound, we evaluate the predicted solubility, and just like for the permeability, the key was how can we shield polar group, not just hydrophobicity and polarity. Here also the key thing is how do we break the lattice, for example, to gain solubility, not just add polarity or reduce hydrophobicity. So that's the dimension that FEP solubility adds in. And it's, again, as we know from MedChem, that's an important design element in order to balance everything.
Yeah, just to add to that we also had some nice machine learning models that Sayan brought in later for things like hepatocyte stability or efflux, right? We had some really nice models, again, as the program had matured and advanced, and we had new challenges with each of the scaffolds. Sayan was able to bring in the appropriate modeling, whether it be physics-based, as he talked about, or some of these machine learning modelings where we had additional data sets, right? And we started applying those and adding those into the MPO scores as well.
So you preempted my next question from the audience, which was, were there any machine learning approaches applied? So
I think Greg answered it.
Yeah, I think I covered some of that. Yeah.
Great. Question around crystal structures that were generated. How important was it to have a crystal structure for a new scaffold?
So it's really critical, I think, as you're bringing forward new chemotypes. So for instance, when you're doing the virtual screen, you find all these very interesting and quite orthogonal chemotypes to what we saw with the FEP core hopping. They could be rapidly optimized using FEP modeling, provided you had high resolution co-crystals. So we did find that to be a critical parameter. So we actually had a collaboration at Nimbus with Proteros in Germany, and they did all of the JH2 crystal structures, TYK2 JH2 crystal structures for us. And we had, quite a fast turnaround from them, and we generated, I would say, upwards of 20 to 30 co-crystal structures, again, all different scaffolds, in order to refine the FEP modeling as the program moved along.
Okay. A question around, did you encounter any interesting water molecules in the on or off target sites, and were those taken into consideration at all?
Yeah, so interesting water molecules are implicitly taken into account. By implicitly, it is automatically taken into account in the FEP calculations.
Okay.
It is explicit waters.
Yep. Does FEP work well when the structural changes are in a highly hydrated region, solvent channel?
Yeah. FEP works very well when changes are in hydrated region and so on. That's taken into account in the rigorous physics of the entropy and enthalpy. It's all good. Yeah.
Okay. How was the off-target binding for other kinases measured? Was it experimental or in silico, or a combination of both?
Yeah, certainly we did both, right? So for the JAK family member off targets, we were looking at both the FEP modeling that Sayan told you about on those off targets, as well as testing that and checking on its activity experimentally in binding assays as well. And then certainly in terms of the larger kinome screens, that was only done on the late stage compounds, like ultimately the development candidate. That was done obviously experimentally in a radio-labeled kinase assay format.
Okay. How did you reconcile any differences between MMGBSA and FEP calculations?
Yeah, so MMGBSA has implicit solvent, first of all, whereas FEP takes into account explicit water molecules, which relates to a question that was asked. So that's number one. And then the entropy and enthalpy are rigorously taken into account both in FEP. So we expect FEP to be way more accurate than MMGBSA, and we see that in fact. The other thing is MMGBSA can be quite series specific or chemotype specific. So you can have situations where locally it's quite is correlating well with the changes observed in binding potency, but when you make large moves across series and so on, the thresholds can change for what's good and what's not good in MMGBSA, whereas FEP is a much more general method. So yeah, so given the choice between the two, I would use FEP every single time.
Yeah, same here. I always liken it. I mean, it got to the point where, given its accuracy, it's essentially become an in silico assay for us on the team. We were, it's highly predictive prospectively of the potencies that we observed experimentally.
Great. Okay, so you mentioned that you filtered thousands of design ideas. Were these ideas mainly coming from de novo design, virtual screening, growing in the binding site?
Yeah, so we use both a lot of hand-drawn designs from Craig, Jeremy, and others, as well as enumerations from time to time. Yeah.
Okay. How well did in vitro potency correlate with in vivo potency for the GH2 domain allosteric compounds?
Yeah, I think Sean mentioned this in the presentation. I think, Sean, correct me if I'm wrong, but it's about 80% of the compounds are within 1 log order, right? For the
I think that, yeah, so that's with FEP versus in vitro. Yeah. I think the question is on translation of in vitro potency to in vivo efficacy.
Oh, okay. I'm sorry, I didn't realize that. I mean, certainly we saw correlations between the binding affinity and the cellular activity, and that correlated well to what we observed in our PKPD models. If we had C-trough coverage of cellular IC50s, we would observe good efficacy in those models.
Okay, good question on the limitations of FEP.
All right. I will give two parts of two-part answer to this. So first of all, for example, if you have large structural changes that is not captured by crystal structures and you're not aware of it, right? And that is a place where if you can see outliers between the FEP predicted potency and the experimental potency. So that's really the major one, I would say. But I'll flip it around, and the second part of it is the advantage of doing rigorous prediction of potency, taking into account everything that is known, is that when there is an outlier, you can use that as a hook to learn something new. So if for given crystal structures, that the potency is predicted to be potent or so on, and you see the outlier, then you can go after that compound to get a crystal structure and try to, expand what you know about the model. Also, there have been several situations across many projects where there was an outlier, and because FEP is so rigorous, we actually looked into the synthetic protocol and figured out that the synthetic protocol needs adjustment because the wrong compound was made, for example. So the advantage of actually using a rigorous method beyond being predictive and making good compounds is that when there is a discrepancy between what's predicted and what's measured, that can actually be used as a hook to learn something new.
Great, thank you. Question around, was there any progress on modeling basic amine-containing molecules charge change? Sayan, maybe you can speak to this.
Yeah. So modeling basic amines is now a standard part of FEP. It is a bit more time consuming for both the modeler and the compute to do that, because you have to model both the neutral and the charge form and then take into account the pKa. But it's now a set protocol on how to do it.
Great. really good question. How high resolution of X-ray is needed to get good results using FEP? Can you use a basic cryo-EM structure greater than 2 angstrom?
Yeah, so a team, joint Nimbus Schrodinger team actually published, I believe, a paper in Nature or something like that, where they used cryo-EM structure with FEP to show that that's quite predictive. So used judiciously and carefully, cryo-EM structures can definitely be used for FEP.
Yeah, I think we used to have, I know with Proteros, we used to tell them that we want at least a 2.5 angstrom or better kind of resolution in order to enable FEP modeling. I don't know if that's still the case, Sam. You could speak to that better than I could, but that was at the time that we were doing this work, that was kind of our cutoff.
Yeah, I think, go ahead, Sam.
Yeah, so I think that's definitely ideal and what we should shoot for. I do think that we can get away with slightly lower resolution if modeler is careful. Yeah.
Yeah, and I would say a huge area of development at Shcrodinger is to enable a broader range of targets for structure-based design and for FEP, a number of new technologies to enable that, to predict poses. And I would say if you have a project you think might be of interest, we have both some interesting technologies and some services offerings as well that can help enable some projects to get FEP running.
Oh, Kate, so to go back to a previous question on the limitation of FEP, Katie mentioned poses. So that's another place where if the pose flips, 180 degree or something, you will see an outlier between the predicted FEP, because that's with a particular pose and the experiment. But then again, you will learn that something has gone awry, that it's either the pose has changed a lot, if there's a hypothesis on that, and those hypotheses can first of all be computationally investigated with absolute FEP with different poses, as well as then going after a crystal structure.
All right, please feel free to put some more questions in. We got a few more here. Did you test different force fields for FEP and MD, or just use one, and which one?
OPLS force field is what we used. I will add that the force field team has been expanding the coverage of OPLS force field, you can find the papers and so on, and the coverage is actually very, very high right now at this point.
Okay. At what scale do you have to run FEP on a project to be impactful, do you think?
Yeah, so I would say for generating new cores, it should be on hundreds to a couple of thousand FEPs. Over the course of a year, I would say it should be about 10,000 molecules through FEP in order to be effective. And on the compute side, that's equivalent, I would say, purely on the compute side, running the FEP, that's equivalent to having one CRO chemist in India or China. That's the effective scale.
Yeah.
so I do want to give some actually a deeper insight on why that is. So one of the on the scale. So one of the things we are trying to figure out, of course, is to develop the SAR in silico. And running, let's say 10,000 ideas, which is, let's say an order of 5 to 10-fold more than what a typical medchem program would make, or something like that, gives you a really expanded view of the SAR. And that really allows one to find interesting directions to go after with actual synthesis, where that can improve how one can gain potency or balance potency and ADME. So essentially, that's how I think of the scale, to do more than what can be done synthetically over the course of a typical project.
Great. And just to get an idea, how long did these calculations take?
Yeah. Again, so that has evolved quite a lot, because there are more GPUs available, GPUs are faster, and so on. These days, these calculations would be an overnight thing. Things were much more constrained back when we were doing the TYK2 program. So we had, of course, but we used a computational strategy to get around it, where we did a single edge potency scan, for example, sometimes on target, followed by full cycle closure and other selectivity and solubility and other calculations, so to get around that. So yeah, so these calculations are pretty reasonable to run these days, and we can always come up with computational strategy to stretch it.
Great. In the last minute here, I'll ask a sort of bigger question, and Craig, also curious your thoughts on this. Where does sort of the physics and FEP and others stand in relation to sort of like AI and maybe, you know, how those two approaches are being combined? Also, Sayan, you can maybe speak to that too.
Sayan, I can take that. Yeah.
All right. So I want to actually frame this with 2 or 3 example scenarios. So for solubility, we talked about it. For example, the lattice is made of the ligand interacting with neighboring ligands, different parts of the ligand. So R1 of ligand can interact with, you know, R3 of the neighboring one ligand and R2 of another ligand, and so on. The point being that the solubility, to actually predict the solubility, you need the whole ligand. It's not just changing one part of it. That leads directly to the chemical complexity around it and the size of the chemical space and all that we are saying. So for the particular program, new chemotypes, new series, you basically need to take into account the whole ligand. And that information is simply not there in the public data sets, for example, because you're going after a complete a situation where it's all possible R1 times all possible R2 times all possible R3, and you soon have an explosion of the chemical space. So that suggests that just learning from the public data set will not have information enough to extrapolate into chemical space, for example, for solubility, as an example. For permeability, similarly, the shielding can happen locally, but it can also happen with other parts of the ligand. So that, again, brings in the chemical complexity and the size of the chemical space. So places like that, where we are extrapolating to new chemotypes, where it's important to take into account the full ligand and the full physics of, is where physics-based modeling is extraordinarily powerful. On the other side of it, you know, there are things like PPB, for plasma protein binding, for example, where we have found that machine learning based methods are superbly effective, whereas physics-based modeling would struggle because of all the number of pockets and things and so on. So that is to say that that it's important to fit the problem to the solution to the problem, and that problem solution fit is very important in how the two work together. So we see it as a complementary scenario, and we have used it as such. For example, Craig and I have done fragment screening that we have talked, in silico fragment screening that we have talked publicly about, where we run FEP for a couple of thousand ligands and so on, then train a machine learning model on the FEP data in order to build a machine learning FEP model that we can efficiently triage then millions of compounds with, millions of in silico compounds with. So essentially the two things are working together. Then, you know, for example, we talked about structures with AlphaFold, that's AI technique, but then those structures feed into what's needed to run FEP, give us the starting point. So once again, a very complementary situation. So this is an evolving situation, obviously, as you know, people are working more and more to figure out where these things fit. But as of now, it's very complementary.
Great. All right, we are at time. Really great questions, and thank you, Craig and Sayan, for taking the time. And
thanks, Katie, for organizing this webinar.
Yeah. All right.
Thanks,
Thanks everyone.